My name is Michelle Doucette, and the date is 4/21. And I am going to do my presentation on determination of gender, genes versus hormones. So to start out with sex determination, I wanted to talk about the sex organs. These consist of gonads, internal genitalia, and external genitalia. The gonads for the male are the testes, which produce sperm, and the gonads for the female are the ovaries, which produce the eggs. And then internal genitalia contain accessory glands and ducts, and the external genitalia are the external reproductive structures. Sexual development is programmed in the human genome. Each nucleated cell in our body, except egg and sperm, have 46 chromosomes. These 46 chromosomes are made up of 22 pairs of autosomal chromosomes, and then there's one pair of sex chromosomes. So as you can see in the picture over here, these are the 22 pairs of autosomal chromosomes, and then this one pair right here is the sex chromosomes. The 22 pairs of autosomal chromosomes are responsible for directing development of the human body form, and then the one pair of sex chromosomes direct development of internal and external sex organs into either male or female. So to go back to those sex chromosomes, that one pair, these can either be an XX or an XY. And as you can see right here, the XY is for male and the XX is for females. Sex chromosomes determine the genetic sex of an individual. And the males who have an XY chromosome get their X from their mother and the Y from their father. And the females who have an XX get both X chromosomes from their mother. The X chromosome is larger than the Y chromosome, and it contains essential genes that are missing from the Y chromosome. So because of this, the male must have at least one X. Sexual differentiation occurs early. So sexual structures begin to differentiate in the 7th week of development. So in the 7th week of pregnancy, you're able to start to differentiate whether or not you are having a male or female baby. Before this point, embryonic tissue is considered bipotential because it cannot be identified as either male or female. So as two possibilities. The bipotential gonads are influenced by a developmental signal which determines whether they develop into testes or ovaries. If the signal is present, then the bipotential gonads will develop into testes, and if it's absent, the bipotential gonads will develop into ovaries. The bipotential internal genitalia consist of two pairs of accessory ducts, the Wolffian ducts and the Mullerian ducts. And as development proceeds, one pair of duct develops and the other degenerates, depending on whether or not the embryo is developing into a male or female. The bipotential external genitalia differentiate into male and female reproductive systems as well. So what determines whether or not zygotes become male or female? Well, sex determination depends on the presence or the absence of that signal I had just discussed, which is the SRY gene, which stands for sex determining region of the Y chromosome. I'm going to move me so you can see the picture. If this gene is present, the bipotential gonads will develop into testes, and then if this is absent, they will develop into ovaries. So this is the SRY gene, and this is on a Y chromosome. So Since it is present, the bipotential gonads would develop into testes. In male development, the SRY gene is present, which causes the development of the gonadal medulla into a testes. Then once the testes are differentiated, they begin to secrete 3 hormones that influence the development of the male internal and external genitalia. The 3 hormones secreted from the testes include the anti-Müllerian hormone, testosterone, and DHT. The anti-Müllerian hormone causes the embryonic Müllerian ducts to regress. So remember, I had said before that the bipotential internal genitalia consist of 2 pairs of accessory ducts, the Wolffian ducts and the Müllerian ducts. So this hormone causes the Müllerian ducts to regress, and then the testosterone that is also secreted from the testes converts the Wolffian ducts into male accessory structures, the epididymis, vas deferens, and seminal vesicles. Additionally, testosterone controls the movement of the testes from the abdomen into the scrotum. And the last hormone that is secreted from the testes is DHT, which is a derivative of testosterone and is responsible for the differentiation of the male external genitalia. So the 3 big hormones secreted from the testes are the anti-Müllerian hormone and the androgens, which consist of testosterone and DHT. Female embryonic development occurs because the SRY gene is not present, causing the bipotential gonad to develop into ovarian tissue. Without the anti-Müllerian hormone, the Müllerian ducts develop into the upper portion of the vagina, uterus, and fallopian tubes. Additionally, because the testes have not been formed, there is no testosterone and DHT secretion. Without the testosterone, the Wolffian ducts will degenerate, and without DHT, the external genitalia take on female characteristics. And that is it. Thank you.